So my name's Melanie Saville, and I'm the Director of Vaccine Research and Development with CEPI. And I'm going to spend some time talking about safety and effectiveness of COVID-19 vaccines, what we know and what we still don't know. So if you move on, first of all, just wanted to introduce you, for those that don't know about us, to CEPI, the Coalition for Epidemic Preparedness Innovations. So we are an organization that was set up in 2017 to accelerate vaccine development to emerging infectious diseases and to ensure equitable access to the vaccines that are developed. And it's here important to note that the importance of working with both the public and private sector here to ensure that vaccines get developed. And so with that in mind, back in early 2020, when the COVID-19 pandemic hit, as an organization that were really set up to respond to emerging infectious diseases, CEPI invested in development of vaccines for COVID-19, really around three main criteria: speed of development. So you can see here, looking at the new rapid response platforms such as the mRNAs, as well as some more validated platforms at the time, viral vectors and proteins that are, although not necessarily the fastest, but are certainly validated. The second particular parameter to look at is scale. So in this case, as we saw very early on, moving into a pandemic, we needed platforms and vaccines that could be scaled to hundreds of millions of doses of vaccine, as this is a global pandemic. The third key parameter is access. So here, in terms of the vaccine development, ensure that vaccines that are developed can be deployed to countries around the world, irrespective of whether they're high-income countries or low-income countries. And another important piece in terms of development, in terms of access, is having a good geographical footprint of manufacturers that can serve all of the globe's community. You can see here, so we have 10 active vaccine development projects. Of those, 2 are amongst the most advanced, with Moderna and AstraZeneca, vaccines that are now licensed and widely used, but other advanced candidates across the various platforms. I will speak quite a lot about variants, and for many, if not all of these candidates, the developers are also working on potential vaccines for new variants. So as we move to look at safety and effectiveness data, I did want to start off by just saying the importance of having the right tools to support R&D. In particular, looking at having standard tools that are available to assess the vaccines so that you have the opportunity to compare. And from the context of safety, this means ensuring that there are good case definitions that people can consistently use across various vaccines to monitor the safety. And when you look at looking at the immune response as a marker for efficacy, the importance of having standard assays that can be used with standard reagents is really critical to really understand how well any particular vaccine may be performing, and also to support potential correlates of protection. In particular, CEPI has a network of centralized laboratories and has also been working with the NIBSC in the UK on an international standard, also with WHO, so that assays can be reported in international units rather than arbitrary units. And these are all important points to consider when looking at what evidence is available for any specific vaccine. Another important point to consider, I already mentioned this, variants. Obviously, as we went through last year, we have a huge number of sequences going into the GISAID database. What is the biological relevance of these to vaccines is a critical point. So again, CEPI has actually set up a task force called Agility, which is bringing together a partnership we have with GISAID on reporting, Public Health England, NIBSC, to look at the biological relevance, looking at cross neutralization from original strains to new variants of the vaccines, and also some early animal data to look at relevant animal data for impact on variants. And as you'll see, there is some evidence that the B1351 from South Africa does have some reduced efficacy. And this is an important point to continue to monitor as we move forward. If we look specifically at safety, what do we know about safety? What are the safety gaps? You will have seen with the vaccines that are being developed that they were developed very rapidly. However, so going from phase 1 all the way through to a phase 2/3 in a very short time frame. In terms of the size of the safety databases, pre-emergency use, they were large, with on average 30,000+ subjects being enrolled in clinical trials, with good safety data being collected. However, obviously the follow-up of the safety data is in the short term. And for example, the FDA requested that at least half of the subjects in the efficacy trials conducted in the US were followed up for 2 months. But as with any vaccine, there are always limits in the number of subjects that are followed up pre-rollout. And one of the interesting observations that has been seen post-rollout of vaccinations is what we're seeing with the adenovirus vectors, where there appears to be a new syndrome associated with thrombosis with thrombocytopenia, which characteristics haven't really been seen before. And this is showing the importance of surveillance post-licensure, post-introduction. So this is a new syndrome. It's being called thrombosis with thrombocytopenia syndrome. The Brighton collaboration have put together an interim case definition so that this can be used consistently for evaluation of potential cases. With this in mind, there is now data both from the AstraZeneca adenovirus vector from chimp adeno and also the Ad26 vaccine from Janssen that cases of this new syndrome are appearing post-vaccination. So it is pointing towards a platform type situation. Where we are today, it appears that roughly 10 in a million cases are observed with the AstraZeneca vaccine and approximately 2 in a million with the Janssen vaccine, but more data is needed. And this slide here, so with that in mind, actually, the regulators, WHO, have reviewed the data on a frequently and have all concluded that the benefit risk assessment remains very positive for vaccination. However, what we have seen with some country NITAGs is that people are putting age restrictions on the use of the AstraZeneca vaccine, as most of the majority of the cases are seen in younger adults and probably more in women than in men. And this illustrates a nice way of looking at the benefit risk assessment according to exposure risk, which resulted, for example, in the UK deciding not to use the AstraZeneca vaccine in the under 30s. And this is in the context that there is a low exposure risk. And in this context, with the low exposure risk, the benefit risk may not be so favorable compared to a medium or high exposure risk. And each country are taking an approach to look at the benefit risk in their countries, in their specific epidemic or pandemic situation. So then maybe look at vaccine efficacy and impact of variants of concern. First of all, in terms of efficacy and effectiveness, we have to recognize that there are many gaps in terms of research for a number of different populations, both from a safety and an efficacy perspective. So a number of populations, we have little or no data in pregnant women, pediatric populations, little data in terms of booster at this point in time, although many vaccine developers are planning boosters and looking at the impact of those. Heterologous primary series or heterologous prime boost regimens are also being looked at, and the safety and the effectiveness of those types of regimens needs to be looked at. Many other aspects, in particular the efficacy against viral shedding and asymptomatic infection and the impact on transmission, are an important factor to overall look at the impact of vaccination specifically. And also obviously looking at the efficacy against the new SARS-CoV-2 variants is going to be important moving forward, and I will show you some of that data. So let's pause just a minute and talk about the variants and variants of concern. So WHO have named 3 variants of concern. There are also a number of variants of interest. And this shows you the growing body of evidence we have for variants as they emerge. And for vaccines in particular, we need to look at evidence of impact on immunogenicity and evidence of impact on efficacy and effectiveness. And you'll see here that there is some data on the variants of concern, and I'll show you some of that data as we move forward. So I want to use the next 3 slides to look at the data that is available on the 3 variants of concern. But first of all, to highlight the efficacy data for non-variants from the efficacy trials that have been conducted so far. So you can see in the 3rd panel here that the efficacy against COVID disease, usually with a case definition with mild, moderate disease, and the majority of the cases being mild and moderate, but in some cases evidence against severe disease. But you're seeing generally good efficacy against the non-variants in the context of the vaccines. Specifically, some of the developers were doing efficacy trials in the UK. So you can see specifically Pfizer-BioNTech, and Novavax, and you're seeing that, in fact, in terms of efficacy or effectiveness, there remains good efficacy against the B.1.1.7 variant, which was first identified in the UK. If you move further to the left, you see impact on neutralization, work done in the laboratories, and you can see there is some reduction in cross-neutralizing antibodies, but relatively moderate reductions, and that probably is reflected in seeing still quite good efficacy. If we move to the next slide, this is the B.1.351 strain, first identified in South Africa. And again, a number of efficacy trials were ongoing in countries, and we have some efficacy data here. And in this case, with AstraZeneca, we have to say this was a small trial and probably was not powered to show efficacy, but there was really little evidence of efficacy for the AstraZeneca vaccine against the B.1.351. You're seeing with Johnson & Johnson and Novavax that there is efficacy, somewhat reduced. There were severe cases with the Johnson & Johnson vaccine, and it appeared that protection against severe disease remains good. However, when you look at the neutralization and cross-neutralization, there is quite a significant reduction in neutralizing antibody titers. So there is some association between the level of neutralizing antibodies and the efficacy observed. In the next slide, This shows you the P1 variant, which was first identified in Brazil. So there's more limited data here, and what you're seeing in terms of the cross-neutralization is similar levels of reduction that you see with the B117 candidate. So a lot of the developers are really focusing on the B1351 variant as one that has potentially reduced efficacy. And then subsequently, if you need a strain change, for example, that at the moment that would be the prime candidate. So what about real world evidence of vaccine effectiveness? So maybe just to start off with some case definitions, efficacy versus effectiveness versus impact. So under development, randomized controlled efficacy trials have been conducted. This is obviously the gold standard for efficacy. But then moving after introduction to effectiveness studies, where you're looking for reduced risk of disease amongst vaccinated individuals in the real world conditions, often observational studies that are non-randomized. And then obviously looking at vaccine impact, and this is looking at the reduction of incidence of disease in a population of which some of the population are vaccinated. So in this case, you're looking at both direct and indirect effects of vaccination. So why do vaccine effectiveness studies? Well, it's very important to continue to evaluate, especially in real world conditions, what the impact, to help understand the impact of vaccination. And it can address a number of gaps that the efficacy trials under clinical trial conditions cannot necessarily meet. This is some specific outcome measures, so much better precision on severe disease, which can be difficult under clinical trial conditions, symptomatic, asymptomatic infections, getting some idea of transmission, for example, household transmission studies, are all important aspects to overall look at the impact of vaccination. Subpopulations, understanding better subpopulations at risk, duration of protection. Obviously, the vaccines that have been developed so far have been shown to be highly efficacious over a short period of time. We need much longer term data, and to understand whether a revaccination is needed, or indeed revaccination with a variant vaccine. These are all critical factors, so really provide important data moving forward. And here, just to show you some of the effectiveness data that has already been generated. First of all, with the Pfizer vaccine in Israel. Israel was one of the first countries to have a very large mass vaccination program. And with such large numbers, you can really look at, for example, when vaccines start to become effective. And here you've got some data post-dose 1 and more than 7 days post-dose 2. So by the time you're getting to greater than 7 days post-dose 2, you have really very highly effective vaccines being demonstrated here in the situation in Israel. If we then move to the US, where data is available, again, looking over time in terms of when vaccines, post-vaccination, the types of efficacy according to time. And again, you see the same thing here with the Pfizer vaccine and Moderna vaccine, where by the time you get to 36 to 42 days after the first dose of vaccine, which will obviously include second doses of vaccine, you're seeing highly effective vaccination. And then this last example shows you Scotland and the rollout in Scotland, where both Pfizer and AstraZeneca vaccines were rolled out. So obviously the Pfizer vaccine and mRNA, and the AstraZeneca as a vectored vaccine. And you can see some potential differences here into time to protection. But again, both vaccines there showing high levels of protection from disease in these examples. So I'm recording this presentation. I think by the time we actually get to the session, there'll probably be an awful lot more data available, but obviously more data will be needed to demonstrate the safety and effectiveness of the vaccines that are being developed. It's important to build the tools to harmonize the assessment of vaccines, so with standard protocols, reagents, etc. Because we will be looking towards, for example, correlates of protection that will be able to be used to demonstrate the effectiveness of vaccines. We've illustrated that safety assessment should be based on the evaluation of the benefit risk assessment. And for this, we need good and consistently used case definitions. And the vaccine efficacy currently supports the continued use of the vaccines, even where there have been very rare safety events, such as the thrombosis with thrombocytopenia syndrome that has now been identified with the adenovirus vectors. And as I say, it's always a situation of looking at benefit risk. And there is also growing real world evidence of effectiveness of the Pfizer-BioNTech vaccine and the AstraZeneca vaccine against symptomatic and severe disease in the adult populations in several countries. So as I said, I think the data will be growing probably day by day, but this gives you a flavor of where we are with COVID-19 vaccine safety and effectiveness. Thank you.